Hello, welcome everyone wherever you are joining us for today's webinar. My name's Alexander Love and I'm a Senior Editor for Global Data. We're very pleased to have you attend today to discuss the topic navigating the complexities of cell and gene therapy logistics. I'd like to welcome you to our webinar platform. This webinar is browser-based, so if you disconnect, please click the link you received via email to rejoin the session. If you'd like to use the Q&A function to ask a question, you can do so using the widget on the platform. We'll try and get through as many of your questions as we can once the discussion has ended. And please use the help widget if you experience any technical difficulties or require any further assistance. And you can use the related content box to access any additional resources. Today's webinar is brought to you by Oximio. These are our guests today. We have speakers Zayheda Khan, Dr. Zayheda Khan, who is Chief Commercial Officer and UK Managing Director. Zayheda has more than 20 years of experience in a variety of roles within the clinical trials sectors, and you can just see on this short bio just the diversity of areas that Zayheda has experience in. Our second speaker is Olga Vizgalova, who is Regional Director for Central Eastern Europe at Oximio. And Olga has more than 15 years project experience of managing projects in the food and clinical trials industry. And she oversees operations and growth strategy, focusing on enhancing Oximio's regional impact. And this is the agenda for today's webinar. So you can see we're going to start off with some introductions to cell and gene therapy, and there'll be some market analysis from global data. Then we'll be delving into the cell and gene logistic challenges, and then looking at overcoming operational challenges, and we'll be rounding things off with a Q&A. So I'd just like to start with some market analysis, as I said. So this is being produced from Global Data's Intelligence Center. And you can see just this curve of this graph, just the vast growth expected by 2030. So the cell and gene therapy market is projected to reach $76 billion by 2030 in terms of market worth. This represents a compound annual growth rate of 44% from 2023 onwards. As illustrated in the chart, the market has grown significantly from just over 2 billion in 2020 to then double in 2023 to reach 5.88 billion. And as you can see, it's forecast to accelerate sharply over the coming years. This surge is driven by advances in personalized medicine, increased investment, and rising demand for curative therapies that are targeting rare and complex diseases. For logistics and clinical supply chain partners such as Oximio, this exponential growth highlights the urgent need for scalable, compliant, and agile infrastructure to support the evolving demands of this innovative therapy landscape. So, as I said, the cell and gene therapy market is not only experiencing rapid growth, but it's also diversifying across different molecule types. So by 2030, within this 76 billion projected worth, the largest share of the market is projected to be gene modified cell therapy that is contributing 44% of the market. This is closely followed by gene therapy at 42%. Let's just have a look at the market size projected by therapy area. So oncology dominates the cell and gene therapy landscape already, but this is only expected to continue in the years ahead up to 2030. So by 2030, the largest share is oncology, which is going to account for projected 51.6%. This reflects the urgent global demand for next generation cancer therapies that offer precision and curative potential. Beyond oncology, you can just have a look down and see the other therapy areas that are also going to be experiencing significant growth. And many of these different therapy areas have larger growth in 2030 than much of the market in 2020. So you can see next down after oncology with 10.6% share of the potential market is metabolic disorders, followed by hematological disorders with 7 0.1% or 5.4 billion. And then we go down to central nervous system disorders, which is projected 6.8% or 5.2 billion. And then as you go down, it becomes more diversified. Now, this diversification of indication highlights the broad applications of cell and gene therapy. It also signals the need for specialized logistics and regulatory planning tailored to the nuances of each therapeutic area, especially as therapies evolve from oncology into chronic and rare diseases. And the numbers speak for themselves. But let's have a look at what cell and gene therapy is and involves. And I'm going to hand you over now to Zayheda.
Thank you, Alex. And I think those numbers are also a stark reminder of obviously the growth in this area through various, like we say, tailor-made personalized medicine therapies. So I thought it would be interesting just to go through to have a quick understanding of what cell and gene therapy actually is. So cell and gene therapy is a form of treatment in which living cells are introduced into a patient to repair or replace damaged tissue or enhance the body's ability to fight diseases. These cells can come from the patient's own body, which is the autologous form of therapy, or from a donor recipient, and that could be the allogenic therapy. And we'll go into a little bit more details of how those are derived in the next few moments. Some of these cells may be genetically modified to improve their therapeutic effectiveness, for example, to better target and destroy cancer cells. So you mentioned, Alex, oncology being the biggest division there, and that's to make sure that the cells can target those cancer cells very sort of in a tailor-made fashion. So the classification, as I've indicated, comes under two categories: autologous, where the cells are actually derived from the patient, and allogenic, where the cells are actually obtained from a donor patient or a donor recipient going into a donor recipient. These cells can then further be unmodified, or then gene-modified cell therapies, where the cells are genetically engineered before they're administered to the patient. And this is where you become very much the tailor-made patient solutions for such treatments. Where you do then have to modify such cells and the gene therapy modifications that do need to take place, that the gene is inserted into the patient cells outside the body and then the cells are modified and then reintroduced into the patient. From an in vivo perspective, the gene is delivered directly into the patient's cells within the body using a vector. So there's a lot of immunological vectors that need to be the delivery vehicle for such treatments. Together, these therapies represent a transformative shift in medicine, offering a highly personalized treatment with the potential to address previously untreatable and rare conditions. Where we look at cell and gene therapy supply management, this slide outlines the end-to-end supply chain for cell and gene therapies, and it is far more complex and sensitive than for traditional pharmaceuticals that we have been handling for many, many years. Each step requires precision, coordination, and strict compliance due to the personalized and time-critical nature of the cell and gene therapy cells, but also the actual scheme and the planning phases. So if we do walk through the process, the main key here is the consultation, the planning, and the expertise that are required to make sure that you have the correct guidance from not only the manufacturing phases, but also then optimizing the timing and ensuring the compliance with regulatory and GMP requirements through the processes. Donor selection and collection. The process also then begins with selecting the right donor. Often the patient themselves in autologous therapies is the donor themselves, and the cells are collected through a clinical procedure, such as a biopsy, for example, and then reintroduced to the recipient from their own body, going back from a collection to their own body. Cryopreservation, so the cells have to be collected and frozen immediately to maintain their integrity during transit and storage. And this is crucial to prevent any degradation or contamination of the cells. Further on from this, the shipping solutions have to be specialized, and logistics providers such as Oximio ourselves have to ensure that the material is frozen from the onset all the way through and shipped under tightly controlled and ultra cold conditions, maintaining full chain of custody and traceability. Manufacturing is a core of this supply chain, and the manufacturing process involves genetic engineering, cell expansion, and product formulation. And each batch, as we've indicated, is tailor-made to that individual patient, so there can be very little room for error. The shipping solutions, going back and from manufacturing to clinical sites, the therapy must be provided and delivered back to and from the clinical site, again under strictly regulated conditions, often with a very narrow delivery window. So the consultation process through every phase of this supply chain has to be critical and undergo quality checks and preparation and consultation phases at every step to ensure readiness and to ensure that the last mile logistics is carefully coordinated and carefully managed. And finally, the therapy has to be administered to the patient. And at this stage, again, timing, product coordination, and traceability have to be critical to ensure successful treatment. So the key messages here that unlike standard pharmaceutical supply chains, cell and gene therapy supply chains are very patient-specific, highly regulated, and time-sensitive. Success does depend on seamless coordination across many stakeholders: clinical manufacturing, clinical sites, biosample management, logistics providers, and also the courier companies themselves. I'd like now to pass on the floor to Olga, who will now go into the storage and logistics solutions that we at Oximio have managed and how we've also overcome some of the challenges that we faced. Olga, over to you.
Thank you, Zayheda. Let me continue with explanation of some logistics aspects of the trials. These slides highlight the key logistical hurdles that companies face when planning and executing cell and gene therapy trials. These therapies are not only cutting edge, but also incredibly complex from a supply chain perspective. Let's walk through each challenge and what they mean in practice. Compliance with regulatory requirements. Cell and gene therapies are governed by stringent and evolving regulatory frameworks. Logistics providers must meet global and local standards such as import and export licenses, chain of custody and identity of protocols, and region-specific documentation, for example, FDA, EMA requirements. Maintaining compliance at every handover point is critical to ensure product approval and patient safety. Unlike standard pharmaceuticals, cell and gene therapies involve living biological materials, often patient-specific. In such therapies, the patient can be both the donor and recipient, and these cells may be irreplaceable. Even minor hurdles, errors, and temperature deviations can compromise viability. This demands end-to-end control management for every phase of the process. Time-critical logistics. Timing is not just important, it can be life-critical since most cell and gene therapies are for patients who are very poor, and such treatments can be the last resort. These therapies follow a tightly synchronized process where each step, collection, manufacturing, quality control, or for example, re-administration, all occur within very strict timelines. As previously indicated, even a small delay can result in a huge failure. Cell and gene therapy logistics aren't just about moving a package. They involve coordinated, validated, and often cross-border operations, including transportation to specialized GMP facilities. Variables such as custom clearance, regional regulations, weather disruptions, infrastructure limitation, all this must be anticipated. Effective cell and gene therapy logistics demands multi-tiered contingency planning. Plan A, B, sometimes even plan C and route flexibility to handle the unexpected. From cryogenic conditions to ultra-cold requirements, cell and gene therapy products must be maintained within the precise temperature ranges throughout the supply chain. Any fluctuation, even brief, can jeopardize product integrity. Temperature control, storage, and transportation need to be monitored throughout the whole process. Given the stakes, real-time visibility is not negotiable. GPS tracking, temperature and shock sensors, and condition-based alerts allow logistics teams to proactively mitigate issues. The transparency builds trust with the stakeholders and enables rapid responses in case of deviation, preserving both the product and the patient outcome. Let's have a look at this in action. So Oximio case study, scalable model in action, it's meeting cryogenic and ultra-cold storage needs for our client. A cell and gene therapy sponsor needed end-to-end logistics support for transporting highly sensitive biological materials and investigational medical products. The challenge included handling multiple temperature regimes. From -20 to 2+8 for biosamples, such as whole blood and tissues, with a very extensive monitoring period. And -196 for IMP requiring cryogenic storage and transportation. What was our scalable solution? Drawing on our flexible and scalable model, we delivered a tailored logistics plan with robust quality control instructions and procedures. For biosamples handling, for materials requiring frozen, deep frozen, we deployed validated and selected packages coupled with real-time temperature monitoring to ensure uninterrupted cold chain integrity. For IMP, LN2 cryogenic logistics. IMP were stored using vapor phase liquid nitrogen tanks, minimizing direct exposure to LN2, while maintaining consistent cryogenic conditions. For transportation, we use certified LN2 cryo shippers, engineered to preserve drug stability from dispatch to delivery. The outcome were safe and compliant transportation. All materials arrived intact, maintained within validated temperature ranges. Cross-border delivery, seamless global transport, meeting all regulatory and safety standards. Regulatory compliance, full adherence to local and international guidelines for handling biological goods and IMPs. Through close collaboration with the local teams and stakeholders, we applied our established protocols and lessons learned to extend operations in Georgia and Israel. This cross-team knowledge sharing enabled rapid setup, aligned standards, and smooth service continuity, providing the strength and agility of our operational model. So, to sum up, at Oximio, our operational model is built around flexibility, global contingency, and local adaptation. We have successfully implemented our cell and gene therapies logistics model across multiple locations, including Ukraine as pilot, and then expanded to Georgia and Israel, providing its ability to scale under diverse regulatory and infrastructure conditions. Cell and gene therapy rely on precision. That's why we enforce globally recognized quality protocols across all the sites. No methods allocation, our process meets the same rigorous standards, ensuring sample integrity and regulatory compliance throughout. These therapies are not delivered in isolation. Our success lies in aligning clinical sites, CROs, laboratories, and logistics teams in real time. Whether managing biosamples in transit or resolving custom delays, proactive communication keeps operations on track. We use specialized equipment. Biological materials can be irreplaceable. We deploy cutting-edge cryogenic containers, validated LN2 storage solution, and robust temperature monitoring system to protect every single product. Equipment is only as good as the people operating it, so we invest in continuous training for our teams. Every minute matters, so we oversee streamlined processes. From collection to delivery, we have built an integrated system that allows for rapid handoffs between stakeholders. Our streamlined workflows allow us to navigate complex supply routes while avoiding bottlenecks and reducing turnaround time. Supporting cell and gene therapies, trials required niche expertise in temperature-sensitive logistics, document handling, regulatory protocols, and risk mitigation. Our teams are multidisciplinary and deeply trained, so clients don't need to worry about the technicalities. Biosamples from sites, very sensitive topic. Collection and transfer biosamples require zero margin to error. We coordinate first mile pickup with advanced tracking and temperature monitoring to ensure samples are preserved from the moment they leave the clinical site to the moment they reach laboratories. Export and import licenses. We don't just move packages, we manage documentation, custom approvals, and international compliance to keep shipment moving. With each country presenting its own legal landscape, our logistic teams are well versed in navigating cross-border permits. Supporting global trials means handling layers of evolving documentation, from GDP, GXP, and customs forms to country-specific ethics requirements. We centralize and localize these processes for clients to ensure speed, safety, and success. So, our model is scalable and agile. It's designed to scale fast and adapt locally. We don't sell infrastructure, we sell capability. Whether a client needs LN2 in Kenya or same-day handoffs in Georgia, we can establish it rapidly. By mapping resources, training, and infrastructure, we can replicate success in new geographies in 2-3 months. Our rollout is modular, allowing us to meet bespoke client needs while ensuring consistency across regions. Scalability is not just about expanding reach, it's about preserving quality and coordination as we grow. We don't need to say we have done it, we show that we can do it anywhere. So why work with Oximio? I will let continue Zayheda. Thank you.
Thank you, Olga. So we talk about obviously our can-do attitude and what are the aspects that you can see here in terms of why Oximio can actually provide the competitive advantages in cell and gene therapy. We offer flexible, tailor-made solutions. We can connect with pharma and patients worldwide to provide such flexible services, tailor-made to make these unique challenges of each clinical study and how we can overcome these challenges of each clinical study, no matter how unique they may be. We provide uncompromising quality. We at Oximio have made a commitment to provide the excellence is backed by advanced processes, infrastructure, and a culture which is dedicated to quality to deliver the best-in-class solutions to the highest standards. Fulfilling the needs of the clinical trials across the globe. As Olga has mentioned, we leverage our experience and expertise to satisfy clinical sites studies worldwide. And if we need to set up and actually advance a certain process in other locations, we can do that in a very rapid manner. And for cell and gene therapy, which usually means around 2 to 3 months in a very fast-paced environment. We elevate our customer experience, we ensure transparency, and we ensure that we have an integrated platform with our customers to ensure that there's a transparent and very clear route of every single step of this of the solutions that we're offering to our clients for their projects. And we cultivate a strong customer relationship. It's essential for us to ensure that we provide swift and effective customer solutions across all organizational levels. It's important to make sure that we can meet your demands of your clinical study. As we reach the end of our webinar, I'd like to thank you all for joining us today. We've hoped that you found this information useful. And there's obviously a lot more in-depth information that we can provide to you. So feel free to reach out to us. But I will now pass over to Alex. And now I believe that we may have a couple of questions that we may be able to answer for you. Alex, over to you.
That's right. Thank you so much, Zayheda and Olga, for your presentations. Some really interesting stats and analysis there. So yeah, we're now going to move on to the Q&A. Just a reminder to the audience to ask any questions. If you'd like to do so, please use the Q&A box on the platform. You can see at the bottom of your screen. We've had some that have come through already, and we'll try and get through as many of these as we can during the rest of this webinar. So I suppose the key takeaways, just before we go into the Q&A, the key takeaways from today's webinar, just about really highlights the complexity of cell and gene therapy. It really requires meticulous planning and flexibility. There's also the critical importance of temperature control and time-sensitive transportation to ensure desired patient outcomes. And then there's finally Oximio's ability to overcome challenges, really demonstrates its expertise and adaptability. Okay, so we have a couple of questions that have come through while you've been talking. So, right, here's the first one. Which countries is Oximio able to support for cell and gene therapy logistics? Olga, is that one you'd like to take?
Thank you, Alex, for the question. We at Oximio has extensive expertise in managing cell and gene therapy logistics and has successfully supported multiple clinical trials. Thanks to our scalable model, we are able to deliver solutions across all countries where Oximio operates and beyond. This includes, but it's not limited, because according to our scalability, we can provide the solution within 2-3 months. But for now, we can extend our current services in Ukraine, Georgia, Israel, to tower locations in Turkey, European Union countries, United Kingdom, Kenya and South Africa, Serbia and Balkan region, so everywhere where Oximio operates and even beyond. We provide full service support from custom clearance, GMP compliance, storage, and real-time temperature monitoring to direct-to-site deliveries. I hope I responded to your questions. Thank you.
Yep, that's great. Thank you, Olga. And the next one kind of leads into this one quite nicely. So what if things don't go to plan?
Maybe I can take this one, Alex, for you. I think as, you know, sometimes things don't go to plan, and that's okay. But it's important to have that, what your contingency is. You just forward think. You can mitigate some risks. You have a backup strategy. And I think, as it was outlined in a slide earlier by Olga, you have to have option A, option B, and sometimes option C. And it's to making sure that before you actually implement any of those, any of that plan, that you have all of those steps in place. We, as I would believe, as experts in this particular field, that we know what it's like when it comes to geopolitical situation, tariff situations, and we have BCP strategies to actually cover and mitigate those risks. But we are focused on every step of the product journey, from not only the temperature control, which is obviously to mitigate the risk and to keep the stability and maintain the products in question, but all the way through to making sure that the import and export processes are as smooth as possible, and there's no delays that are happening at those steps. So from that perspective, it's making sure that your plan is very much outlined in detail, making sure that as part of that risk mitigation, you do have option A and option B and potentially option C as well, to proactively think that if something should go wrong, then, you know, Oximio can actually maneuver around that situation.
So it's really all about being prepared for anything.
Absolutely.
Mm-hmm. Okay, that's great. Well, thank you both for answering those questions. And I just have some housekeeping just to close out the webinar. So thank you, Zayheda and Olga, for your presentations again today. It's been really interesting speaking with both of you. And to the attendees, thank you for joining us. Tomorrow you will receive an email to say how you can access the on-demand version of this webinar, or you can access it through the website arenainternational.com/events. And please use the related content box to access any additional resources. And if you'd like to continue asking questions, you can do so by interacting with the widgets, as the on-demand version of this webinar will remain interactive. The speakers can provide you with a written response directly, as well as those who have kindly submitted questions today, but unfortunately we didn't have time for. Thank you once again, and I hope to see you on another webinar.
Thank you.